then that will have to be designing.
Too much. Yeah. Great. I'll just,
Maybe take a few minutes because
yeah.
I have a spring break.
Oh, yeah.
They may be still lost. Yeah, it's fine. Wait a few minutes.
Exactly.
Let's get started. Okay. Welcome back from spring break. Welcome back. I'm glad you made it back and not lost in some who knows where it is in spring break. Good morning. Welcome everyone. And my name is Ed Guo. And to my speaker, Guo Xiangdong, and the Department of Biomedical Chair at the Columbia University. So before I introduce our speaker, I have a few instructions to give. Today, we continue to offer this lecture in a hybrid format with both virtual and live audience. So our speaker will present approximately 45 minutes, and we will leave 15 minutes at the end for question and answers. So for our virtual audience, throughout the presentation, please use the Q&A features at the bottom of your screen. And maybe in the future, we should remove this one. So if you do not know about the Q&A, I think you probably should not join a Zoom conference. You should know that now. And for a live audience, you should be able to have a chance to ask a question and bring the question. So today is really my fantastic honor and the pleasure to welcome Professor David Umulis, and his Chinese name is Yu Dao Wei. So his Chinese actually is very, very good. And then we connected on WeChat, and then he replied to my invitation by Chinese. So that's, and then also I only know David for less than 2 years. We met in the Council of Chairs meeting by BMES, I think maybe last year or 2 years ago. And then I introduced myself and said, okay, well, come to visit us at Columbia. But as this moment now, he's really kind of become acting chair already in less than 2 years, but become the senior vice provost of Purdue University. It's really fast trajectory going up. So currently, he's the acting head and professor of London School of Biomedical Engineering at Purdue University and director of NSF EMBRIO Institute. Umulis leads London School to advance discovery and translation in biomedical imaging, neuroengineering, instrumentation, the computational biomedicine, engineering biomaterials, and this NSF's EMBRIO Institute using engineering, imaging, AI, and organismal biology to uncover mechanisms of wound healing and cellular defense. So Umulis is a member of the Purdue Teaching Academy. He has received the 2011 Richard Coles Early Career Award and the Henry Young Award for Leadership for Service. We talked about the Henry yesterday. And give a welcome to Yu Dao Wei.
Thank you. Thank you very much. And let me make sure that I get this shared on Zoom now. So one moment. Share screen. So, are we all good online? All right. Oh. So thanks again for this invitation. I'm really honored to be here at Columbia University. When I was an undergraduate student at the University of Michigan, your outgoing president was the president of Michigan at that time, and I got to see him a few times before before he came here. And then that, from what I understand, that he's wrapping up after 21 years, which is quite a presidential term. So today I want to have this be interactive. I like pauses. Oh, thank you very much. And questions and flow. I'm really passionate about teaching and making sure that students, interact and understand. So part of the talk is about research that, as the outcomes of a method, and I'm really pitching method in this talk about the usefulness of mathematical simulations as part of a tool in the tool set that we use to ask questions and address hypotheses. So instead of a math model coming in to sweep up after the parade, after everything's already known, there's ways to use it rigorously to help us figure out biology. And that's where I think there's value. So focusing on this in the terms of bone morphogenetic protein patterning in embryonic and cochlear development. I'd like to start with an abstract and a proposition. So let's see. Okay, let's see if this was later. It's not as fine. So in a Wall Street Journal article in 2013, E.O. Wilson, biologist from Harvard, had an op-ed to make the case that biologists do not really need to learn any mathematics. And whenever they run into difficulty with numerical issues, they can find a technician or a mathematician to help them out of their difficulty. And he formalized this with Wilson's principle number 1. It's far easier for scientists to acquire needed collaboration for mathematicians and statisticians than it is for mathematicians and statisticians to find scientists able to make use of their equations. Now, that created some interest from me, considering that this is kind of where my career is going, and so I had to think of some responses. But I come from a different background in chemical engineering, which was really emerged from the application of mathematical principles to chemistry in order to scale up processes and things. And there was one of the founding figure in chemical engineering used to quip, "All experiments ever do is ruin a good theory." So, but actually, the truth is somewhere in the middle between these perspectives. And you don't have to look any further than developmental biology to see how mathematical thinking has really influenced developmental thinking and developmental biology overall. In fact, 2 of the highest cited papers in developmental biology come from folks with very mathematical backgrounds, including Alan Turing, who's well known for everything else that he did, but he in biology, in 1952 paper talked about Turing pattern instabilities, where you could have spontaneous emergence of patterns in form where the homogeneous diffuse state is actually unstable. And he coined the term morphogenesis in this landmark paper in 52. Extremely hard to read and understand. But there's been a whole body of work following on this. Henry Hess actually showed me some things that are reminiscent of this, but more in the Rayleigh-Bernard cell reaction. But also Lewis Wolpert formalized thoughts of positional information. So how do you have information that is stored in genetic material, give rise to spatial patterns, and then how do you have responses to that in a quantitative way? The focus of the talk for the first part is going to be on zebrafish development and the development during a process called epibole. So early in development, let's see the pointer here. The zebrafish, after fertilization, there's several rounds of synchronous cell division where there's a formation of a mass of cells that are part of the embryo, and then there's during the blastula gastrula transition, there is the generation and secretion of molecules called morphogens that then form spatial patterns over the developing embryo that are then later subdivided further and further as these cells and tissues are going through the process of morphogenesis and moving physically around the embryo. 24 hours, you have something that starts to look like a fish with a beating heart and structures of a fish. And zebrafish is a very famous system because of how easy it is to image and to follow it with imaging techniques. It's transparent, and you can see these processes play out in a very reproducible way. What is important to the talk today is the patterns that are formed by the morphogenetic proteins that differentiate cells along the ventral to dorsal axis. And I'll provide a little bit more information on what BMPs are next. So bone morphogenetic proteins are secreted cytokines that transmit their positional information by binding to and forming complexes with receptors via the interaction showed here. The process gives rise to a signal, a phosphorylated signal downstream that is the readout that you can actually measure in the experiments that you do to look at the patterns. Where the BMP ligand receptor interaction takes place is where the signaling happens, and this is how you can have a graded amount of the ligand lead to a graded distribution of the signal and differentiation at multiple different domains. Because this interaction is so critical to development, it's very tightly controlled by several secreted factors that really control the access of that ligand to bind to receptors, and they control it over space and time in these systems. All of the signaling in this pathway is done by obligate tetrameric complexes, and the signaling is highest once it forms a heterotetrameric complex composed of two type 2 receptors, a heterodimer ligand in the middle, and two type 1 receptors. And some of the manifestations of mutations in this pathway in development lead to craniofacial developmental disorders, but also with respect to ACVR1, it's shown to be the cause of fibrodysplasia ossificans progressiva, which is a rare disease where wounding induces ectopic bone growth, and it continues over time. So what are the goals of the work within this signaling system, and how do we use models to help us with our studies and hypotheses? We're interested in quantifying the BMPs, the BMP signaling, target gene expression. Engineers like input-output mapping. And one of the first equations I learned when I entered an undergrad was in minus out plus generation minus consumption equals accumulation. And that's basically the only principle that we're applying here, but just in a way where we're measuring inputs and outputs and using a model in between. We want to link genotype to phenotype by determining and simulating mechanism and back and forth. We want to predict outcomes across species via AI approaches to improve power of preclinical models with rigorous tools. And we want to also link mechanics and signaling to predict morphogenesis defects, identify tissue patterning, organization, and precision. And the last part of the talk is a fun new extension in the development of the inner ear. So this is motivated by how does the pattern form in the embryo? And there are many different ways that you can form a non-uniform distribution over space. One possible way is that you just have a poorly diffusive regulator or inhibitor, and that binds up all of your active ligand. And this creates a net flux that would drive a pattern by just adsorption at one end of a domain. Another option is that you have a non-diffusing signal, and then you have an inhibitor come in and form a counter gradient to block access to receptors. Another way that was popular and interesting in Drosophila is that there's a more complex network that can actually use inhibitors to force a net flow of the ligand against their concentration driving gradient away from their source and redistribute them in that way. And yes, a question. Not stupid question.
Fish don't have bones, right? So why is it
So bone morphogenetic proteins, they got their name from the initial indication of what they did in cell culture, their ability to induce bone formation, but it's a ubiquitous, fish have bones, but you're right, the Drosophila do not. It's ubiquitous signaling molecule that does that and many, many other things. It comes from, you find the molecule, you get to give it the name, leads to a lot of creative names, specifically in Drosophila, like noodle spätzle, bazooka, wishful thinking, and others. So, but both morphogenic proteins are potent signaling molecules in the TGF growth factor beta family of cytokines, and they're involved frequently in regulating cell differentiation decisions or maintaining stem cells in niche states and others, but also for their role in bone. But so how does that one mechanism work? I made a little animation to help explain that for the students. So this is actually in fruit fly. The readout there is the top-down view, and this is a cross-sectional view of a fly embryo, where there's a thin fluid-filled space surrounding a syncytium, or all of the nuclei are embedded at the periphery. That's one of the main signaling BMP ligands that are secreted in space, that then that will transmit the spatial information, like I mentioned earlier, by binding to their receptors, which forms the heterotetrameric receptor complex. This initiates the readout that we use in the systems today in the inner ear, the zebrafish, and in Drosophila of phosphorylated SMADs. But to form the actual pattern and explain shuttling in the picture before, in the Drosophila, these secreted inhibitors are also at play, and they're produced in different regions. And at the interface, these inhibitors are known to interact and bind and form more complex and more potent inhibitors that are then really effective in preventing the ligand from binding to receptors, preventing them from signaling on the flanking regions in this particular instance. This is where the protease comes in. There's a protease that specifically in Drosophila, not as specifically in zebrafish, targets one of the inhibitors, SOG or cordon, when it's in this complex with the ligand. And this is an homage to my past here, where the protease destroys cleaves the inhibitor, releasing the ligand. And then the idea is that this process plays out and leads to the spatial localization and distribution of where signaling is occurring. But heretofore, when we started this project, we didn't know what was happening in zebrafish. And there was some indication of what was happening in frogs and xenopus. Less was known yet about what's happening in mouse. And so we wanted to address the question, how is this forming? Let me check this. Is there a question in the chat? Oh, okay. I'll close that. I'll let them know if there's questions. Okay. So what we wanted to do, because it had the same players in Drosophila as we have in zebrafish predominantly, was look at how the pattern is forming, how it's controlled. And so here's a network diagram of the interactions that are taking place, like the ring structure. This is if you look at the top down, it would be a ring structure, but here we have a high signal ventrally, a low signal dorsally, and it is controlled by secreted factors, the inhibitors, noggin, follistatin, TSG, cordon, tolate, and others. And so I also wanted, this is a picture from a study by Berndt et al. That shows the cell movement during the process of that patterning that's taking place. So these are simultaneous coupled events. Let's see. There we go. So Joe Zinski, in my collaboration with Dr. Mary Mullins at University of Pennsylvania, is a co-advised student who worked a lot on developing the quantitative methods and simulations for the first part of the study. And what you see here is a segmented zebrafish embryo where every single cell has been imaged, segmented, and identified for the level of BMP signaling in that particular cell at that point in space. And so what we have here is high-level signaling ventrally and then low-level signaling dorsally in a very quantitative, reproducible way with this method. So you take the imaging, you can go run it through the programs, and you can get the distribution of 8,000 to 10,000 cells, knowing what every cell signaling state is in that embryo. You can do, once you have that method, then you just turn the crank many, many times, do a lot of imaging, get statistical numbers, and you can start to determine how the dynamics of the gradient is forming over time. And so what you see at the ring here, which is shown here in red of this embryo, as a function of time, the signal ventrally increases, but there's a large region on the flanks where the signal is largely stationary and stable. But that's the output. So how do we know how the output is happening? How do we get to that shape of distribution? In order to do this well, we need to quantify the inputs. And so the inputs in the system are, where are things being produced? Where are things being secreted? So Xu Wang and my lab used at the time what were relatively new methods for single molecule mRNA detection, because the ligand expression is very, very low, very, very hard to detect. So using RNA scope and cryosections, she was able to quantify where the main ligand was being produced and secreted in the cross section. So this is a top-down view, a slice of the embryo, and you can see that it starts with a graded distribution that looks like this. And so this is the input of where the ligand's being produced. Other methods were used to quantify, and I'll show you in the next slides just how good these methods have evolved. So here's a in toto imaging of mRNA quantification for the inputs in this particular pathway of where the ligand for BMP is being produced, and also where the protease, the Pac-Man, was being produced, and then where the inhibitors are being produced. So you can see the cells. And then if I zoom in on these individual boxes, the red box and the yellow box, you can see full-length mRNA transcripts around the cells. And then you can also, when there's paused transcription, you can see two spots per nucleus, which is the instantaneous readout of where things are zipping off of the gene and have that level of resolution. But we're more interested in the full-length particles that are in the cytoplasm being translated. So this gives us a distribution over space in these embryos of where the molecules are being produced that we can then use in our model. We had a student in the group who now works at an image analysis company, develop several image segmentation for nuclei, for nascent mRNA in the nuclei, and then for counting of material mRNA around the embryo. So it becomes a fun data problem, a little bit like Google Earth of an embryo, where you zoom out, see overall patterns, you can zoom in and see spots. Finally, Shu Wang was able to develop a method for simultaneous input-output, so we can measure phospho-SMAD signaling, a protein assay, along with mRNA at the same time, and it makes nice images, but you can really see quantitatively the distributions of where these molecules are being produced. So what do you use to bridge the inputs and outputs? This is where the hypothesis is. You're trying to figure out how do you get the outputs based on the inputs, and we formalize the hypothesis with simulations and equations. And the way that we do that is providing the molecular interactions in the simplest form become a series of partial differential equations for in minus out plus generation minus consumption equals accumulation. So for instance, this is an example for the ligand. You have the change in the ligand over time depends on the processes of where it's produced and where it's decayed, as well as the processes for its transport. The initial model we consider just diffusion, later models we consider advection and diffusion. There we have several knowns, and we have several unknowns, and the unknowns is where the hypothesis is encoded. So with the unknowns, you allow freedom, and you will do a screen over the unknowns to identify where what type of process is actually leading to the observation. So we developed a classification logic based on our new computational screen of these models, where we would look at, does the model fit the observed distribution of the p-SMAD, of the signaling? Does the output work? If the answer is yes, how is it happening? Is it happening by the shuttling method, by the transcriptional method? I'll show what those are in a second here. See, is it? Yeah, right here. So is it happening by the counter gradient example that I showed pictorially earlier? Is it happening by a transcriptional method where the input is just directly mapping to the output? Is it happening by shuttling, like the cartoon that I showed you? Or is it happening by the source sink mechanism, where there's some role for an adsorber at one end to create a boundary condition to drive the gradient formation? So what we did was we simulated those equations over 40 million times. In the end, after the second paper, the first paper was about 6 million. And the solutions are based on this sorting. And then we're like, okay, we have really good quantitative data for the inputs. We have really good quantitative data for the outputs. So we should be able to know by fitting the inputs to outputs what's happening. So we compared all these mechanisms and we looked at all of the different solutions. And with just the wild type data and dynamic data, we had many solutions that fit, many positive hits. The problem was all of them worked. All of the hypotheses worked. None of them were eliminated a priori. So the source sink mechanism worked, counter gradient mechanism worked, the transcriptional mechanism worked, shuttling worked. And so it was a lot of work for no gain at this point. And so we're like, okay, well, maybe we can figure out what experiment we want to do next. So we can do biophysical measurements of the binding and transport and things. It takes a lot of time. You have to develop fusion proteins, and then you have to go in and do the imaging, et cetera, et cetera. But when we looked at these biophysical constants and we colored them based on mechanism, we saw there was some distribution, but there was overlap in every single pair of things we would measure. So this told us, even if we did the physical measurement, we really wouldn't have helped our case any. We would still not eliminate mechanism. So what Jozinski did in Mary Mullen's lab was go back and look at several mutants that were already out there and available that have some defects in patterning, but had not been quantified before. And so looking at one of, cordon heterozygous, and looking at the other, some of the mutants, there was no change in signal, no phenotype. It was a negative result. The fun thing is, if you know that it's involved in the pathway, you know that it's a molecule that interacts and binds, a negative result can be used in a numerical screen, because if your system is sensitive to that, then it's not right. So anyways, what he found was that early in development, there's not a big difference in the pattern, but in this mutant with the inhibitor, but later on in development, Jozinski found that there is a large difference in the level of the ventral signaling. So when you couple this data along with some other of the mutants, you can quantify exactly where the differences are. And then you can simulate the mutant conditions and the wild type conditions in the same model and require the model to fit your mutant data and your dynamic data. So it becomes a multi-objective optimization problem. When you did this, we could eliminate 2 out of the 4 mechanisms. By including the mutant data, we were able to eliminate the transcriptional only mechanism. We were able to eliminate the Drosophila mechanism, the shuttling mechanism. There were no situations where shuttling, yeah, go ahead.
How sensitive is this to the confidence that you've identified every factor that's involved in the mechanism? If there's some other binding partner that just hasn't been discovered yet.
So, without changes to that, it would be somewhere baked into the initial or perhaps into what the parameters are. But we do try to not overfit and not underfit. So we can use a model to address the question we're interested in, even if we have an incomplete system, because everybody always has an incomplete system. And it's no different than having a logic hypothesis versus a rigorous testable hypothesis. But here we can have a little bit more confidence in what we eliminate in that. But the unknown unknowns will always get you. So here, what was really good was that when we looked at the decay rate of the ligand and then the diffusion rate of the ligand, we had the 2 mechanisms that were left, source sink mechanism and the counter gradient mechanism. So 2 that could fit all of the data, but they were separated kind of like in a phase space between the 2 mechanism types. So the red dots here are counter gradient, the blue dots here are source sink mechanisms, the gray dots are just the distribution of the screen. And so now it tells us, if we can go in and measure these biophysical parameters, we'll put a crosshairs on this diagram and we'll know definitively which mechanism is consistent. Since these, they phase separate, I like to say, but it's not really phase, but it's closest thing. So then Yabu in my lab went and developed fusion proteins and the protocols to measure the biophysical components. So we did first this fluorescent recovery after photobleaching to measure the fusion parameter for this fusion. And he did it in a way that you'd have clones of cells secreting the molecule we're interested in, clones of cells that did not. So we weren't going to have any concerns about mixing production with transport. And what he found was a very reproducible diffusion rate of 4.4 microns squared per second. And the diffusion rate didn't change with or without the presence of the inhibitor. So that was another test of the shuttling. And this was a control of that. And so this eliminated our simulation outputs to this group. It was source sink in blue or counter gradient in red. And then this narrowed it down further. And he was working on the process of doing the photoconvertible activation to measure decay. And another lab was also measuring decay. I didn't have confidence in our measurements, even though it was indicating a rate, but the other lab had developed a different method and found the rate in a parallel paper of 4 times 10 to the minus 5 per second. So it put it right here. So the crosshairs was right where I'm putting this arrowhead right here. If you can see that, which means that we can now pretty confidently say that the mechanism is the source sink mechanism. It is not the shuttling mechanism or the frequently talked about hypothesis of counter gradient or transcriptional mechanism. But we use the model and a back and forth to help identify what experiments, what measurements, because it took 6 months to do the fluorescent recovery after photobleaching experiment for us to do all the controls and everything else. And so it takes time to do those, and you don't want to do them if they're not going to really help you along the path. But using the model and the back and forth, it really did help us to narrow in and identify the mechanisms that are at play. So this is where things are at now. We are using whole embryo finite element modeling simulations that are built off of the cell velocities and tracing. We have asked now addressing questions of advection of so ligands diffuse. So what we found was that ligands diffuse is not mRNA. The source sink mechanism is consistent with the diffusion. There's no shuttling. So that was a recap of this first part of that part of the talk. We've had subsequent papers and follow-up on it, looking more at the properties with Mary Mullins and Francesca Toison and Jozinski and their group, looking at toluid and other molecules. This back and forth approach has been very helpful to rigorously kind of show how, if you have a toolbox that includes some simulation, you can actually do new discovery. So the most recent work that we've just completed, there's a preprint available now in bioRxiv, still the same molecules, but a completely different context and system. So we wanted to, I read this paper in development that came out in 2014 or 15 that really made me interested in knowing the role of bone morphogenetic proteins in the precision of patterning of the inner hair cells in the developing inner ear, in the cochlea. This is the main mechanosensory neural center that is the hair cells that detect the vibrations in the sound waves after they process and flow through the outer and middle ear and transmit that information into the nerve pulses that we interpret as sound and language. So we started this project with a PhD student, Matthew Thompson, and our collaborator, Vidya Munamalai, who's a scientist, research faculty at Jackson Labs. And we were interested in, this is incredibly precise. In all of our studies of the BMP signaling, it doesn't seem to be as precise as what is showing here. So what's going on? And motivating this work was the fact that neurosensory hearing loss is something that is very, it's pervasive. It increases with age. My niece got this when she was 5 after one round of a type of influenza, and in mammalian systems, the hair cells in the neurosensory tissues don't normally regrow, and so. Just a quick overview of the biology of the system. We have the outer ear, the middle ear, the inner ear that ultimately transmits, amplifies, processes the information that allows us to hear in this cascade for converting analog signal and conduction into the sensor neural, which is more of a digital signal as an output based on the pulsing of the neurons and the interpretation in the brain. But if there's hearing loss, what happens in sensorineural hearing loss, you end up having a breakdown in the final step, where either by loss of the cells or other types of reasons that you're unable to, to take the information from the analog to digital conversion. Jumping ahead a little bit, there's one of the things that we're really interested in with BMPs is regeneration, stem cell control, and growth. And in some systems, the hair cells do regrow back, but in humans they don't. And in other mammalian model systems, they don't regenerate. we could find a way to reactivate those pathways, it could really make a dent in sensorineural hearing loss. So the chick and zebrafish are examples that regenerate in several contexts, but the mouse is a system where it does not. And so in the early development paper that was really inspiring to me, there was some studies that showed how small disruptions, very small disruptions in the BMP signaling would cause that beautiful pattern of hair cells to get all misaligned and have very different numbers and lead to defects. So zooming in more on the inner ear of where this is all taking place, there's a spiral structure in the cochlea. And if you were to take a cross-sectional view of that and zoom in on one of the twists of the cochlea, the main sensorineural organ is in this organ called the organ of corti. This is where the hair cells ultimately are. And this is where that developmental picture I showed you of where they are. So it was more complex than the examples I just showed you for bone morphogenetic protein signaling. There are several morphogen families that are working at the same time, including Notch, FGF, Wnt, and BMP signaling, as well as Sonic the Hedgehog, which is frequent in these types of systems that show this level of precision. This is a very nice review article from Vidya Munamalai and Donna Fekete in 2019 that kind of outlines all that's known about these integrative morphogen pathways that ultimately control and give rise to the position of the structures in the developing cochlea. What is interesting to me is that if we start to look at the outputs and try to structure this up in an input-output mapping, like I showed you earlier, we find that the BMP pathway has outputs like PMED that we already showed in zebrafish, and that turns out the reagents basically work the same way. But a readout of the other pathways is a transcription factor called SOX2. And so we focused on having SOX2 represent what's happening from these FGF, Notch, and Wnt pathways, and we focused on BMP simultaneously to see if it was enough information from SOX2 and BMP signaling that could provide precision. So Matthew Thompson worked with Vidya to develop a way to do the quantification of SOX2 and PMED signaling in sections of developing mouse cochlea. And he quantified these distributions, much the same processes that we would use in the zebrafish embryos. And what you see from embryonic day 12.5 through 14.5 in both SOX2 and PMED signaling, the SOX2 signal changes quite a bit over time on this normalized distribution, where there's quite a dynamic shift. Phosphosmed signaling, the BMP signaling, basically stays in lockstep and persists for 2 days. The gradient is stable for 2 days. What's also very interesting, and I'll talk a little bit more about it in a little bit, is that this gradient doesn't look like any gradient that I've seen before. And I've studied a lot of gradients. This is, I'll show evidence of this. It's a straight line, it's linear. So if you look at these in different ways, you can plot what the SOX2 expression looks like on one day versus the other. And this just quantifies how much change there is, how much dynamics there is over space in what SOX2 is doing. And all this says is that, yeah, PMED is pretty stable. SOX2 really is shifting over time. When we tried to fit several different functional forms to the shape of the PMED gradient, we found that the fit that worked the best at all the time points was the equation for a line, y equals mx plus b. Knowing physics, knowing transport, knowing reaction and transport, it's not so easy to get a straight line. Normally you'd get an exponential shape just by the processes of diffusion and reaction. So this was intriguing to me. But it was also intriguing to me because we've been studying, I'm going to jump ahead one slide and come back to this one. We'd been studying these papers about interpretation, positional error, and position in readouts of a morphogen gradient. And this one came out in genetics from Thomas Greger, Petkova, Gaspertasic's group, in Gaspertasic in, I think, 2015. And they had just a purely theoretical study looking at how do you end up with precision in morphogen patterning. And so they looked at shapes of gradients, of just putative gradients. And they found that the information maximizing shape of a gradient occurs when the gradient is a straight line and doesn't have the curvature or is binary. And they quantify this information content with bits, which is the binary 2 to the n, I'll go back a slide now to show what our measurements are. But the bottom line of this study was like the boring linear gradient, which I haven't seen before, is the one that has the most information. But there was a theoretical paper that showed a linear gradient, and it was in Nature in 1970 from Francis Crick. And it was a paper called Diffusion in Embryogenesis. And this is a figure from the paper showing how putatively you could get a linear gradient. The math is easy, but actually having that happen in biology is hard. But there was a precedent now for a linear gradient, and also a precedent that a linear gradient is information maximizing. So we went and we started to calculate the number of bits to have a number of domains. We would need to have 8 domains to specify precision of the regions of the inner ear development. And so when we have PSMAD 159 by itself, the linear gradient, we already have 2 bits of information, or the ability to specify with precision, for 4.6 domains. But when you combine it and have a joint distribution of mutual information with SOX2, the joint distribution gives rise to 8 domains that persists for 12.5 and 13.5, which means that theoretically, just by measuring those gradients, there's enough information content in those gradients to have enough precision to specify the different cell types in that tissue. We have a lot more work to do in that system, but I'm not sure, that we'll be able to do it because we graduated, and so the mouse part of my lab is now kind of stopped. But it was really interesting to take some of these observations and then see this surprising linear gradient. We did a lot of simulations to see how to give rise. The only thing that gives rise to a linear gradient is a source sink. And that's exactly what we just did in zebrafish. And other mechanisms don't make a linear gradient. So what we found is that the source sink is the only mechanism that produced linear gradient in simulations. The first observation of prediction made by Francis Crick in 1970. So we're the first ones that I can find in literature to have found a linear morphogen gradient. And it seems specifically capable of providing the precision needed for inner ear development. So that was a really fun observation. So I talked a lot about these folks throughout my talk. I didn't go in too much into the Drosophila work, which was what Tembi Maduli had done a lot, but want to thank the collaborators that have been working a lot with us over the years. Mary Mullins' lab at University of Pennsylvania with the grad students Joe Zinski and Francesca Toisan. Matthew Thompson did the inner ear work. Yabu did all the work for the fluorescent recovery after photo bleaching. Xu Wang did a lot of simulations and also the quantification of mRNA. Adrian Buganza has been helping us a lot with the 3-dimensional finite element modeling. Linlin Li has been the primary modeler for the BMP signaling and patterns. And then Wei Dou and George Wu did a lot of the image analysis and initial studies. And then Vidya Munamalai is the colleague at Jackson Labs. So lastly, I want to finish with a little plug. These ideas of the back and forth between modeling and experiment, we decided to go after a large NSF institute, and we put a team together to go after problems of wound closure and the coupling of signaling with mechanics, and the coupling of lots of experimentation with lots of image analysis and modeling. And we landed at NSF NSF Biointegration Institute, and we have an active summer training workshop. Last year we had 50 students participate. We're going to have it again this upcoming year, where a lot of the methods that we talk about here are taught in a course setting. And we're also continuously looking for more postdocs and participants for the Embryo Institute activities and workshops. I want to thank you very much for your time. I want to thank very much Ed for the invitation to come here. I've greatly enjoyed getting to meet the faculty, and I look forward to meeting students this afternoon. So thank you.
Yeah, somebody will be beginning to control. Thanks so much for very fantastic presentation. And before we go into the questions, I just want to call everyone's attention to the next hybrid seminar, which will take place next week on Friday, March 31st. Stephanie George from National Science Foundation is going to talk about navigating and hopefully receiving BME related fundings from NSF. That's a good. Hopefully many of our faculty will show up, and those inspiring future faculties can also get to know how to get money from NSF. So now, and also please follow us on social media to receive the latest update, and you can see our social media information on these slides, and you always access registration information for our seminars. So that's why, Mr. David, we have a script, because most of the hosts without script will skip the propaganda material to that meeting. So
No
now,
worries.
yeah, I'm gonna turn to Lisa to handling the questions and answers. Yeah.
So we have a couple of questions online, but we like to start with student questions first or from the faculty. Any questions up there? Well, I oh, good.
So the Francis Crick model with the linear distributions has boundary conditions that are really ideal of like a source that holds the fixed concentration.
Yeah.
And the sinks of the source.
Yeah.
All work in.
Which doesn't happen.
Yeah, so biologically, how would you envision that approximating that system? So you think it's just coincidence?
So the easiest way to have you need two things. If you have a sharp boundary of a perfect adsorber, it can basically ensure the one condition at one end, where you just have the concentration zero. At the production end, I don't know if this is at play, but the only way to really get a constant concentration source easily is if there's a signal reception at the source and a feedback, an automatic feedback loop to attenuate it, kind of like a proportional controller. But I don't know if that exists either. But yeah.
Actually, one of the questions from the online audience relates to that, just in case it's possible to explain why the linear gradient is the information maximizing.
Yeah, so there's trade-offs in the distribution of, so you have normally you measure the error based on the y-axis or the signal difference at a position. And then you map that to a positional error, and they do a good job of it in that TASIC paper in 2015. And the trade-offs would favor the linear gradient for the entire domain, but not for every single position along the domain. So the other examples that showed the kind of the curved one at the source and where it plateaus at near zero, you have very, very high positional error there, because the delta X for any small change in the signal, delta Y, you move a lot in the X direction for the errors in those domains. Whereas if it's a straight line, it's just the compromise that balances both of those.
So another follow-up. Did the Thomas Greger paper talk about how you get it, or they just put it out there as this would be best?
That paper was theoretical, and they focused on shapes, but not really the mechanisms that give rise to the shapes. Yep.
Okay, so now another one here. Maybe you can read it.
Oh, sure.
I'll read it out loud too, but thank you for the talk, David. For the first part of the talk, BMP diffusion and early zebrafish development. Regarding that, could you comment on the role of cell density in regulating the rate of morphogene diffusion, since the embryo is continuously dividing? Do you think the embryo is able to sense the number of cells at a given time and adjust the geometry of the morphogene rate?
That's a really thoughtful question. And it's one that I've actually thought about a lot because there's some transport theory that looks at transport around and in and around obstacles. And what's interesting about the early stages of development, as the cells divide, they do get smaller. And so the paths might be a little bit more tortuous, but we didn't see any differences in their rates of transport. But from a theoretical perspective, I wrote a very, very detailed paper about that exact question, and it was in Royal Society Interface. I think it's the analysis of dynamic morphogen scaling or something like that, that looks at exactly that question that you're asking right there, and looks at all the different limits of the cell packing density and that role that it has on the pattern itself as it's changing over time.
Well, I have a question and a comment. I really like the inner part because that's my field, but and I also really like that you're studying how the organ develops, because people are so focused on hair cell by itself, like hair cell regeneration, as though if you could regenerate hair cells, you'd be good to go, but really you need this whole pattern.
Yeah.
So it's really nice work. It seemed like the interesting stuff you were studying was between like embryonic day 12 to 14. Is that right?
Yeah.
Okay, so is that when that pattern is established?
Yeah,
Okay.
that's when it's a lot of changes. That's some folks have had success in growing them in vitro, ex vivo, but this was all these were all sections of cochlea from embryonic tissues. I'll try to get the reference. Everybody's interested for the paper that motivated. I should have put that in there, but I haven't talked about the cochlea stuff very much. We have one preprint available. Happy to hear more questions about it. I always like, iron sharpens iron. So this is a new direction for us, but it's really fascinating. I really enjoyed the science of it.
So I think there's one more, but it's like, what was the x-axis on slide 23? So I'll let you do that one.
Sure.
And then otherwise, are there any other questions from the audience?
I think it's gonna still recovery.
That's okay.
So, all right, well, let's thank our speaker one more time.
Thank you.
Those complication simulations, so you just run them each time they will have characteristic and have different solutions.
Yeah.
So how long it takes generally?